What are the 10 biotechnology?
1. Animal agriculture. 2. Energy and environment. 3. Food innovation. 4. Fermentation technology. 5. Diagnostics. 6. Healthcare pharmaceutical. 7. Genetic engineering. 8. Plant agriculture crop improvement. 9. Genomics proteomics. 10. Bioinformatics.
One of biotechnology is animal agriculture. It is the practice of breeding animals for the production of animal products and for recreational purposes. It is discovered in the Neolithic Revolution around 10,000 years ago, but may have begun much earlier. It has been speculated that human beings used fire to cook food 1.5 million years ago, but the only archaeological evidence obtained thus foresets the date of the use of fire for cooking at 12,500 years ago, as indicated by the discovery of clay cooking pots in East Asia and Mesopotamia. Neolithic tribes took the first steps towards agriculture, discovering that some plants could be grown from seeds and some animals can be tamed.
Environmental biotechnology is the solving of environmental problems through the application of biotechnology. It is a biotechnology that is applied to use and to study the nature environment. Environmental biotechnology could also simply that want to try the hardest biological process for commercial process and exploitation. The International Society for Environmental Biotechnology defines environmental technology as a development, use, and regulation of biological system for recommendation and contaminated environment such as the land, air, and water, and the environment-friendly processes and the green manufacturing technologies and sustainable development. Environmental biotechnology can simply be described as the optimal use of the nature in the form of plants, animals, bacteria, fungi, and algae to produce renewable energy, food, and nutrients in a synergetic integrated cycle of profit-making processes where the waste of each process becomes the feedstock for another process. The replacement of fossil fuels with more carbon-neutral and renewable sources has become a key necessity of the time. The proportion that energy can be obtained from biomass with a decisively positive energy balance and at a scale is sufficiently large to have a substantial impact on sustainability and security objective is both supported by several recent studies. Developing a sustainable economy more extensively based on renewable, carbon, and eco-efficient bioprocess is one of the key strategic challenges for the 21st century. However, the potential social, economic, and environmental impacts of biofuels have been the subject of the controversy.
Hungarian engineer Carl Erigi discovered energy and environmental biotechnology in 1919. It is utilized in waste treatment and population control, and it can clean up various wastes more efficiently than traditional methods, reducing our reliance on land-based disposal methods dramatically. Especially, it is refers to the use of procedures to safeguard and restore the environment quality. And it can employed in the variety of methods to detect, prevent, and remediate toxic emissions into the environment.
In the early 18th century, Frenchman Nicolas Appert developed a method for preserving food in glass jars. Aptly known as the father of canning, Appert also is considered by some as the father of food science. Innovation in the food industry combines technological innovations with social and cultural innovations. It occurs throughout the entire food system, including production, harvesting, primary and secondary processing, manufacturing, and distribution. The ultimate innovation is a new or improved consumer's product and service. innovation can be focused in one area of food technology, for example, process engineering, product formulation, food qualities and consumers' needs, but ripple spread causing changes in other parts of the food system in consumers. Eating patterns in general social and cultural areas. Food industry innovation strategies need to be based on the total technology in the food system and consider not only with the technological changes but also with the social and environmental changes, so as to produce food that satisfies the nutritional, personal, and social needs and wants of all communities. It is vital to hospitality industry because businesses must compete with new ideas in order to meet the constantly changing needs of customers. Food innovation is the development and commoditization of new food products, processes, and services. Right now, it's happening rapidly.
Fermentation technology. By the way, this is Rico J. Paigao Spanyola of Bachelor of Science and Tourism Management 1C2. Fermentation technology. It is the anaerobic conversion of sugar to carbon dioxide and alcohol by yeast or any group of chemical reactions induced by living or non-living ferments that split complex organic compounds into relatively simple substances. In fermentation, a chemical change is brought on by the action of microscopic yeast, molds, and bacteria. Fermentation is the process involving the biochemical activity of organisms during their growth, development, reproduction, even senescence and death. Fermentation technology is the use of organisms to produce food, pharmaceuticals, and alcoholic beverages on a large-scale industrial basis. The basic principle involved in the industrial fermentation technology is that organisms are grown under suitable conditions by providing raw materials, meeting all the necessary requirements such as carbon, nitrogen, salts, trace elements, and vitamins. The end product formed as a result of their metabolism during their lifespan are released into the media, which are extracted for use by human beings and that have a high commercial value. The field of fermentation technology has been the science of many stormy developments in the past decade. The major products of fermentation technology produced economically on a large-scale industrial basis are wine, beer, cider, vinegar, ethanol, cheese, hormones, antibiotics, complete proteins, enzymes, and other useful products. Fermentation technology was discovered by Louis Pasteur. The turning point came when Louis Pasteur, during the 1850s and 1860s, repeated sequence experiments and showed fermentation is initiated by living organisms in a series of investigations. In 1857, Pasteur showed lactic acid fermentation is caused by living organism. And that would be all. Thank you.
Diagnostics technology. Biotech diagnostic tools use genetic information to help doctors detect and diagnose conditions faster and with greater ease and accuracy. Doctors can now tailor disease prevention and treatment to individuals' genetic information. Many of these diagnostic tools are now portable, allowing physicians to conduct tests, interpret results, and determine treatment on the spot. These tools have had a profound effect on the access to healthcare in developing countries where the healthcare infrastructure often undeveloped.
My name is Karen Vanyas from this Team 1C2, and now we're going to discuss one of the biotechnology, which is the healthcare or pharmaceutical. The pharmaceutical industry discovers, develops, produces, and markets drugs or pharmaceutical pharmaceutical drugs for use as medication to administer to patients with the aim to cure them, vaccinate them, or alleviate the symptoms. The first modern pharmaceutical drugs was invented by Friedrich Sturner, a German scientist, in 1804 when he first extracted the main active chemical from opium and named it morphine after the Greek god of sleep.
My name is Janlidia Kina from BSDM 1C2. Similarly, while GlaxoSmithKline's origins can be traced back as far as 715, it was only in the middle of the 19th century that lithium became involved in the industrial production of medicine, producing patented medicine from 1842 and the world's first factory for producing only medicines in 1859. The main goal of the pharmaceutical industry is to provide drugs that prevent infection, maintain health, and cure diseases. This industry directly affects the global population, so a number of international regulatory bodies monitor things like drug safety, patents, quality, and pricing.
Genetic engineering based on recombination was pioneered in 1973 by American biochemists Stanley N. Cohen and Herbert W. Boyer, who were among the first to cut DNA into fragments, rejoin different fragments, and insert the new genes into E. coli bacteria which then reproduce. Genetic engineering is the process of using recombinant DNA or rDNA technology to alter the genetic makeup of an organism. Traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offspring with desired traits. Genetic engineering involves the direct manipulation of one or more genes. Most often, a gene from another species is added to an organism's genome to give it a desired phenotype.
Originally, the phrase genetic engineering applied to a variety of approaches for altering or manipulating organisms through the processes of heredity and reproduction. As a result, the term encompassed both artificial selection and biological procedures, interventions, including as a artificial insemination, in vitro fertilization, test tube infants, cloning, and gene manipulation. Many theoretical and practical aspects of gene function and organization have been improved thanks to genetic engineering. Bacteria capable of manufacturing human insulin, human growth hormone, alpha interferon, hepatitis B vaccine, and other therapeutically important compounds have been developed using recombinant DNA technology. Plants can be genetically modified to fix nitrogen.
Crop improvement refers to the ways of developing and breeding of crop varieties which are better than the pre-existing cultivars in a number of characters like taste, height, color, etc. for a better yield. It is essential to meet the demands of changing world, example increased population, climate change, and decreasing land base. Plant breeding programs are continuously striving to increase crop yield, enhance crop quality, and improve crop tolerance to diseases and pests. How crops can be improved? Crop genetic improvement by GM or conventional approaches. It is only one of many methods that can be used to improve crop performance. Other involve improvements in farm practices, irrigation, drainage, and herbicide, pesticide, and fertilizer use. How can we do genetic improvement of crop plants? The foundation of Mendelian genetics enabled plant breeders to cross plants with a new precisions, carefully manipulating the plant's genome to produce new, improved varieties. These breeding techniques have been used to develop higher yielding varieties, including plants' resistance to pests or disease. What are the aim of crop improvement? The objective of crop yield improvement are higher yield. This can be done by the process of hybridization and crossbreeding, better quality. There are different reasons for improving the quality of different crops, such as improvement of the baking quality in cereals like wheat.
Who discovered? Mark Wilkins. The first studies of proteins that could be regarded as proteomics began in 1975, after the introduction of the two-dimensional gel and mapping of the proteins from the bacterium Escherichia coli. When was genomics invented? Although the word genomic is rather recent, its origin goes back to the beginning of the last century, when Johansen introduced in 1909 the concept of the gene, the physical entity of the corresponds to the genetic determinant of an inheritable trait in an organism.
The uses of proteomics and genomics. Proteomics complements genomics and is useful when scientists want to test their hypotheses that are based on genes, even though all cells of a multicellular organism have the same set of genes. The set of proteins produced in different tissues is different and dependent on gene expression. Then it is the purposes of genomics and proteomics. Like genomics, proteomics aims to study the entire repertoire of proteins within an organism. Proteomics is far less advanced than the field of genomics because robust technologies to study the structure and prevalence of all proteins in a cell in a high throughput manner are only now being fully developed.
So hello everyone, my name is Shirley Sermatos. So today I'm going to discuss to you about bioinformatics. So apart from analysis of genome sequence data, bioinformatics is now being used for a vast array of other important tasks including analysis of gene variation and expression, analysis and prediction of gene encoding, structures and functions, predictions and detections of gene regulation networks, simulations environment. Bioinformatics is by nature a multidisciplinary field that began in the 1960s with the efforts of Margaret Odenhoff, Margaret Fitch, Russell F. Doolittle, and others, and has matured into a fully developed discipline. There exists a number of applications of bioinformatics for accelerating research in the area of biotechnology. The term bioinformatics was invented by Colleen and Ben Hesper in the 1970s to study the informative process in biotech systems.
Hi, my name is Rihanna Marie Pogoni from BSTM 1. Pauline Hagerweg, historically the term bioinformatics did not mean what it means today. Pauline Hagerweg and her Ben Hesper coined it in 1970 to refer to the study of information process in biotic systems. Bioinformatics uses computer programs for a variety of applications including determining gene and protein functions, establishing evolutionary relationships, and predicting the three-dimensional shapes of proteins. The field of bioinformatics has three main objectives: to organize vast streams of molecular biology data in an efficient manner, to develop tools that aid in the analysis of such data, to interpret the results, accuracy, and meaningfully. Bioinformatics is defined as the application of tools of